Abe is associate professor for neurosurgery and neurology at Johns Hopkins, just next door. And Abe is also leading the biomarker core, where he has really access, to a wide range of proteomic technology and non-proteomic technology, looking at biomarker monitoring and discovery. And it will be great to hear from his perspective what new technology could be useful in a real-world scenario as a clinician. Thanks, Abe.
Thanks, Yuan, for that introduction. Can you guys hear me? So I'm going to transition from bench to the bedside, and then later on in the talk, back to the bench. I'm going to talk mainly about Alzheimer's disease biomarkers as it relates to my core interest. Many of you may be aware, one of the holy grails, we now have an FDA-approved blood biomarker for Alzheimer's disease. And it's as reliable as PET scans or CSF analysis. And it's essentially the ratio of p-tau217 to A-beta-42 with very high positive and negative predictive value. And while it's an immunoassay and it's not a mass spectrometry assay, the groundwork for this was actually laid by mass spectrometry. Beta amyloid was isolated from the brains of patients with Alzheimer's disease and Down syndrome in the early 80s. And subsequently, a decade later, phosphorylation sites on tau were all identified by mass spectrometry. And I know there are many tools recently, and we are tempted, to do Olink and SomaLogic. You wouldn't, if you had done Olink and SomaLogic on CSF or blood for Alzheimer's disease, you wouldn't have identified the A-beta-42 fragment, which is a peptide fragment, or the p-tau217, which is a PTM. So this tells us that mass spectrometry is still very important despite all these new other tools that are there on the market. And more recently, people have come up with a synaptic signature for Alzheimer's disease. This is a very large cohort. 3,400 CSF samples over 6 cohorts. I mean, really large study. And they did SomaLogic, and they identified 2 proteins, one going down, NPTX2, and one going up, YWAG. And just the combination of those 2, gave you a very high predictive value of who's going to develop Alzheimer's disease. And these are both synaptic proteins. And it took them, that many samples over that many cohorts. But we forget, we described this 6 years ago in 40 very well-characterized samples using mass spectrometry. This tells us you don't need to do thousands of samples to get some real biologic answers. If you have well-characterized cohorts, you can get real biology understanding by just looking with mass spectrometry. So we identified NPTX2 and 1433 gamma, which is the same protein as YWAG. And again, one is going down, one is going up. And we showed that this combination of NPTX2 and YWAG or NPTX2 and PKM gave a very high AUC. Just to show you that mass spectrometry has done this already before. And so now to step back a little about understanding why Alzheimer's disease and what's the role of biomarkers in Alzheimer's disease. The condition was first described over a century ago, but it was not until the 1980s when beta amyloid was first isolated, again, using mass spectrometry from AD brains. Subsequently, tau was purified, and only later was APP, the protein precursor to the beta amyloid fragment, was identified. In the 90s, ApoE4 was identified as the single biggest risk factor for Alzheimer's disease. Subsequently, familial AD mutations were identified in APP and presenilin. P-tau and A-beta-42 was first measured in CSF. The first FDA-approved PET scan was in 2012. It took almost a decade later for the first CSF A-beta-42-40 ratio to be approved by the FDA. The Pressivity C2 and AD2 assay is actually a lab-developed test. It's not quite an FDA-approved test yet, but that is based on mass spectrometry. And again, this is a most recent FDA-approved immunoassay. And this is, the classic amyloid hypothesis of Alzheimer's disease, that sometime in your 50s and 60s, you start developing amyloid plaques in the brain in the extracellular compartment. that elicits a neuroinflammatory response to try and clear out these amyloid plaques. But if this neuroinflammation persists, you get phosphorylation of tau because many kinases get activated. These tau tangles form. And subsequently, you have neuronal and synaptic loss. And that's when you manifest the symptoms of dementia. And this whole cascade can take anywhere from 15 to 20 years. And what is hypothesized, based on at least cross-sectional data, so if this green line represents cognitive symptoms, MRI and FDG PET starts to become abnormal about 5 years before. CSF tau starts to go up 5 years before that, so 10 years before symptoms on average. Amyloid PET about 15 years before, and CSF A-beta 42 starts changing 20 years before. So this is a very long disease incubation period, and it happens over 20, 25 years. And now there are many medications that have tried to and remove these plaques. These are either to the fibrils or to oligomers of A-beta 42, and 2 of them are now FDA approved, donanemab and lecanemab, which is here, yeah. So these 2 are FDA approved. But all models are wrong, and only some are useful. There are problems with these anti-amyloid drugs. So in fact, there is a clear sex effect in these drugs. So they don't benefit females, for example, at all. For ApoE4 carriers, there's a very high risk of edema and hemorrhage and adverse effects. So if you're homozygous for E4, many hospitals will not give you these anti-amyloid drugs. And more importantly, cognition continues to decline. So it doesn't cure Alzheimer's disease. It just slows the progression down. So in a clinic, when I'm seeing a patient and I'm telling them you should take this drug, and my patient asks me, so will I feel better? And I have to say, honestly, no, you are still going to decline. So to the patient, it is actually meaningless because they are still declining over time. And even the benefit is not clinically significant. Yes, you do a large enough study, you'll get a statistical significance, but it's not clinically significant. And subgroup analysis, as in this study, showed that women don't benefit much at all compared to males. So amyloid is not the answer by itself. Yes, it plays an important role, especially in those ApoE4 or people with familial, autosomal dominant models of AD, but in late onset sporadic AD, it's not just amyloid. And we know that because many studies have shown that the pathology is multifactorial. There's many things going wrong in the development of Alzheimer's disease, and just looking at one thing, just looking at amyloid here is not going to be enough. So essentially, we need biomarkers for all these different processes that are going wrong. And there have been some efforts to address this. Again, this is a study using SomaLogic, again, just published a few months ago. Again, large cohorts, and they identified around 15 proteins, some going down, some going up. And again, another recent study that showed that brain-derived proteins in plasma can be detected quite reliably now. So we can infer what's happening in the brain by analyzing plasma. And in a recent editorial in Cell, Carlos Cruchaga laid out very well, how doing these large-scale blood proteomics, not just using the immunoassay or aptamer platforms, but using mass spectrometry, can help give us, organ-specific proteomic signatures, and not just biomarkers, but help us understand disease pathogenesis, identify drug targets. So I think we are heading into a new golden age. To pivot a little to aging, we forget that it's not ApoE4 that's the single biggest risk factor for Alzheimer's disease. It's actually aging. So if you're 65, your risk of Alzheimer's disease is 5%. By the time you're 85, your risk of disease is 40%. So age is the single biggest risk factor for Alzheimer's disease. So if you have to understand Alzheimer's disease, we have to understand what's happening with aging. So we looked at, again, small study, but very well-characterized cohort. Getting CSF from young normal patients is quite challenging. And getting old normals who are completely disease-free, have no vascular disease or Alzheimer's disease, again, very, very difficult. So we looked at this small cohort. They had no evidence of Alzheimer's disease even by biomarkers. We made sure that they are not asymptomatic Alzheimer's disease on their way to progression in the future. Normal cognition with MoCA and the CDR sum of boxes. And this was a study we did about a couple of years ago in partnership with Biognosys. So for plasma, we did do depletion of the abundant proteins and FAMES DIMS. We identified around 3,000 proteins. In CSF, we did neat CSF, no depletion or enrichment, and we got around 5,500 proteins. So we wanted to understand what's happening with aging in plasma and what's happening with aging in the brain by using CSF. And this is looking at the differential expression of the cohort, both at the protein level and at the peptide level. And COMP, this protein that's enriched in cartilage but also expressed in the brain, was elevated both at the protein and peptide level. And looking at the pathways affected, you can see that cell adhesion and immune response pathways are elevated, and neuropeptide signaling pathways are downregulated. Just with aging, again, not Alzheimer's disease. Looking at in plasma, LTBP2 was elevated both at the protein and peptide level, and many others too. And again, looking at the pathway enrichment, we saw, again, inflammation, cell adhesion, up with aging. And vesicular transport and vesicular turnover, microtubule function was downregulated in plasma. LTBP2 has been well described in many other studies as a risk factor for Alzheimer's, for all-cause dementia, including Alzheimer's disease. So I'm not going to go into details of that. We did a WGC analysis to figure out which modules are correlated with age, sex, and cognition. And in plasma, not surprisingly, it's been well known that insulin signaling is affected as we get older. And there's a clear link between growth hormones and insulin signaling, and essentially growth hormone secretion stops around 60. So the single biggest module that correlated with age in our WGC analysis was IGF pathways. This was in plasma with aging. But in CSF, multiple different modules correlated with age, which tells us that the brain is aging at a much different rate than the rest of your body, and many more pathways are affected. So many extracellular matrix remodeling modules are correlated with brain aging. Many of the collagens, some upregulated, some downregulated. Lipoprotein metabolism, again, very important with relationship to risk for Alzheimer's disease, is affected with aging in the brain. Nearly 25% of your cholesterol metabolism happens in the brain, so it's not surprising. And not surprising, again, coagulation and inflammation are elevated in aging, suggesting some blood-brain barrier dysfunction and a neuroinflammatory response just with aging. Because we did mass spectrometry, so we could also look at phosphorylated and non-phosphorylated peptides, and we wanted to look at differentially phosphorylated, non-phosphorylated peptides of the same protein. So with aging, some collagen 4A2 peptides are downregulated and some are upregulated. Same thing for transferrin, NRCAM, and many other proteins, many of them known proteins associated with Alzheimer's disease. So this is trying to, again, get to the link between how aging increases your risk further of Alzheimer's disease. Importantly, ApoE. So ApoE, we found it to be differentially phosphorylated, and it's more phosphorylated at this particular site, and that's very close to the site where it binds to amyloid. So in essence, in aging, we saw upregulation of all these pathways in CSF. In plasma, we saw insulin signaling was downregulated, and we observed over 20 differentially abundant phosphopeptides in many of the proteins that are known to be risk factors closely related to Alzheimer's disease. And now to pivot back a little, yes, amyloid deposition happens in your 50s, but what is triggering this amyloid deposition? What is happening even before amyloid gets deposited in your brain? And again, just to recapitulate, I'm just going to repeat, not go through the whole slide, but these are all the problems with amyloid drugs. So we wanted to look at what's happening even before amyloid gets deposited, because many pathways are getting simultaneously affected along with amyloid deposition, but what's triggering all those pathways? Again, we want to figure out, are there any biomarkers related or pathways related to all these different pathogenesis that we could detect? So we have been lucky to have this BioCard study that we've been following longitudinally for over 30 years now. It's a very well-characterized cohort. These were all cognitively normal individuals that were enrolled in their mid-50s. So it's a relatively young cohort when it comes to aging studies. These are middle-aged people, essentially. And they've been followed now close to 30 years now. The first patient was enrolled in 1995. And they are very deeply phenotyped. They have blood and CSF collection. Blood is collected every year. CSF and MRIs are done every other year. And recently, we also started doing amyloid and tau PET scans. Imagine this. You're a normal cognitive person, and you're volunteering to have a spinal tap done on you every other year for the rest of your life. And eventually, they agree to brain donation. So it's a very incredibly altruistic cohort. And while there are many studies of Alzheimer's disease, most of them are cross-sectional. But if you really want to understand pathogenesis and timing, what comes first, what comes second, you need to do these well-characterized, well-phenotyped longitudinal cohorts. So the study was at NIH for the first 10 years. There was an administrative break for around 4 years over here, and then the study was reconstituted at Hopkins. And this break was because there was an investigator at NIH that was selling samples to pharma and was put in jail. So important lesson, follow your IRB, don't skip. So originally, there were 359 participants. We excluded people who contributed CSF only once. And in all the other subgroups, as you can imagine, of the remaining 240 subjects left now in the study, now, over 25 years, some have progressed to MCI or dementia, but as you can see, the majority of the cohort is still normal. So because now they are in their mid-70s, and it's now that they are transitioning into cognitive impairment due to Alzheimer's disease. And you can see the mean number of CSF collections on average for the overall cohort is 4.5 CSF samples over these 20 years. Interval between the first and last CSF collection is around 18 years. So again, very long longitudinal study. And again, just to recapitulate, so we wanted to look at people who converted to becoming amyloid positive and people who remained amyloid negative over these 20 years. So there were around 53 participants who converted from amyloid negative to amyloid positive, and we selected from the remaining controls, around 52 age and sex matched controls who had remained amyloid negative and were followed for an equally long period of time. This is just to show you the ratio of CSF amyloid. Paradoxically, as amyloid gets built up in the brain, there's less A beta 42 in the CSF as it forms oligomers and plaques in the brain, so less of it comes into the CSF. So amyloid levels in CSF actually drop. So these are the people who progressed. As you can see, the majority of the cohort over this time period is still normal. And these are people with, again, the progressors whose p-tau levels are going up, over the same cohort. Again, the majority of the cohort over this time period is still normal. So what we wanted to look at is the converters versus non-converters. We wanted to compare their baseline, but also look at what's happening before and after amyloid conversion to understand what pathways are affected. And we want to look at both plasma and CSF in these converters and non-converters. Again, we did propensity matching to make sure they are all age and sex and interval matched. The only thing we couldn't match was for ApoE4, which is, again, not surprising. People with ApoE4 are more likely to progress to Alzheimer's disease than people who are ApoE2 or ApoE3. And we had, again, a choice of platforms on which to do it on. We had already done the gold standard, at least core ADCSF biomarkers in this entire cohort. And then we had a choice of figuring out which platform we wanted to use. And we chose, again, to go with mass spectrometry because of the advantages it gives us in terms of identifying isoforms and PTMs, which none of the other platforms will give us. And just to give you an idea, when we did the aging analysis, originally in partnership with Biognosys, 153 samples took us around 5 weeks. And now in the current cohort, we've done the plasma analysis. We haven't finished the CSF analysis yet. It took 2 weeks, with the higher throughput workflow and P2 enrichment process. So this is the workflow for the P2, and Johan could answer questions about that. And on average, we saw around 7,000 protein groups in this cohort over those 500 plus samples. And just to give you an idea, there's some overlap of these proteins with the Olink panel, for example. We are planning to do Olink analysis also in the same cohort. But you get many more unique proteins in the mass spectrometry workflow. And these proteins belong to a wide variety of different pathways, a lot related to inflammation, not surprisingly, but amyloid and tau pathways, synaptic function, blood-brain barrier, again, insulin and IGF signaling. So many pathways that we are really interested in. Again, a median of 7,000 protein groups. And we looked at, how does time of collection affect? So again, some of these samples are from 1989, and we didn't see any separation by a year of collection, which tells you how well-preserved and reliable these measures are going to be over 30 years. We looked at potential pre-analytical contaminants in the time period of this whole cohort across all these 500 samples. Other than just a couple of outliers, most of the samples are of uniform quality. We looked at a few proteins just to get an idea of what's the difference. Again, this is in plasma, not CSF, of converters versus non-converters, and we don't see any glaring fluctuations in expression level over time. But we are still in the process of bioinformatics analysis to, again, do the pathway level analysis to really figure out which are the proteins that are differential in the converters versus non-converters. This data was just acquired, I would say, less than a month ago. So we haven't quite yet finished the bioinformatics analysis, but I'm very curious to see, again, what's different in the converters versus non-converters at baseline, and what changes over time in people who convert to amyloid positivity. So again, all models are wrong, some are useful, and we can improve these models, again, by deeper phenotyping, by deeper characterization of their proteomes, looking at protein isoforms, looking at PTMs. And longitudinal studies are very critical because we have to disentangle what's happening with aging and what's happening with pathology. With that, I'm going to thank many of my collaborators at Biognosys, without whom we couldn't have done this work. Collaborate Mike at Duke, who helped with some of the earlier analysis, and my team at Hopkins. Obviously, NIH, whose funding has been very critical to keep this study going over 30 years. Again, this is the kind of studies that are very difficult to do without NIH funding. And finally, to the BioCut cohort. Again, this is a very unique cohort who has agreed to have such deep phenotyping done on them over the rest of their life, essentially. And we have around 41 of these subjects from the cohort that have passed away and have donated their brain. So we're looking into doing spatial transcriptomics, proteomics now, to really understanding what is the origin of these proteins, which cells in the brain are expressing these proteins. Are they neurons, microglia, astrocytes, oligodendroglia, and all of that. With that, thank you, everyone.